I'm Dr. Jane Thomas. I'm a plant pathologist at the NIAB in Cambridge. The project has been going very well. We've had a lot of samples that have been sent in from growers. Alternaria solani is very characteristic indeed. It produces long beaked spores which are very distinctive. We're also finding a lot of Alternaria alternata, and that produces short club-shaped spores which are much smaller than Alternaria solani and don't have a long beak or tail on them. We see alternata on most of the samples that we receive, or possibly one or two other species, Alternaria tenuis and Alternaria infectoria, which we think are just secondary, but that's part of the studentship as well, looking at the interaction of the species when they're both inoculated onto a leaf. It is more or less what we expected to see, but what we don't understand is how important these so-called secondary Alternaria species are, whether they're really a part of the disease complex or whether Alternaria solani is the main species involved. The interesting thing is that Alternaria solani seems to come in relatively late in the season. So from the end of July onwards, it starts to increase and really becomes more prevalent in September. I think Alternaria has increased in frequency over possibly the last 4 to 5 years. It's certainly become more of a problem in continental Europe, and it seems to be on the increase in the UK as well. The next steps are to do some more field work with some inoculations again at different growth stages and look at the development of the disease, but also to do a lot of controlled environment work where we put different species on detached leaves and then on whole plants and look at the rate of lesion development. And we're also collecting a lot of epidemiological information from the field, looking at weather parameters and connecting those with the rate of disease development that we see in our field plots. For 2017, we'll be looking at further field experimentation, but looking also much more at the genotypic structure of the species that we find in the field and looking at variability both within a species as well as the different species that are involved and seeing whether there is any relationship between that and their ability to infect potato leaves.